on that? Yep, okay. We can actually add more information based on our annotation file. And I was hoping that this would give me chromosome location, because you can do that and it's really cool. Here's what I think is, what I find is that you find hotspots in the chromosomes where all these changes are happening. You know, the thing when you start analyzing these data, you realize that, you know, it's just not numbers. Like, there's even location. You know, there's, you know, that the biggest constraint to gene expression is the actual compression of the DNA itself. It has nothing, you know, promoters and all that stuff, that plays a role, but it's actually how easy is it for the transcriptional stuff to get in there. What you find is when you open up a certain region, you get leakage. And so what you'll find is you'll have a gene that's really high expressed, and then you'll have some on the sides that are actually kind of expressed too. It's crazy. And no one's really taking that into account. And I don't have enough mathematical skill to do it, so hopefully I can get a student that's really good at math. Okay, so here's what I want you to do is we can add more information. So go to probe set ID, right click, and go insert annotation. Now we can start putting in anything we want here. Here's what I want to put in. I want to put in the biological process. So if a biological process is known, associated with that gene, it's going to give it where it is, cellular component, and then it's just kind of molecular function. Is it a kinase? Is it a cytokine? What is it? Oh, and pathway. Put a pathway in there. I have no idea what that's going to produce. Pathway, and hit OK. This is quick and dirty bioinformatics, biological interpretation. Anything similar? I haven't even looked at this, so I don't know. But again, this will give you an idea, you know, what this gene is, right? You know, right now it's just a bunch of letters with a 3 at the end. This is telling you, oh, it's involved in protein binding. And it's in the extracellular space. Come on. And it's in multicellular organism development. What are we looking at? Stem cells, right? They're differentiating. Makes sense, right? I want you to see these. Oh, and in utero embryonic. Look at this. Embryonic development. This isn't just random noise, guys. This means stuff. Like the genes we're pulling out, if I look at what it actually does, it makes sense. That's why I like seeing the genes. I don't like just looking at numbers. If I look at this, I'm like, oh yeah, phagocytosis. You're destroying cells. You're getting rid of some cells as it differentiates. All of this stuff kind of makes sense. When I see this, this makes me feel good. And, and that's why I think if you come with a biological background to this, it's so much better. You can just kind of, be assured that you probably did it right because it does make biological sense. All right? But what I'm going to do next time is what we are going to do is we're going to take these gene lists, make sure you save all your samples. Don't save this yet. So this one I think is filtered, right? Your original one. Go to filters and go clear column filters. You should see the column 35,000 again. Now you can save it if you want. I don't think we did anything to it, did we? Oh, we changed the day. So go ahead and save that. Any questions so far?